Hello there. This begins part 2 of chapter 3. Specifically, this reviews sections 3.3 on nucleic acids, 3.4 on carbohydrates, and 3.5 on lipids. Part 1 introduced organic molecules and talked about proteins. So let's get started here with nucleic acids. First, let me talk about the title. Nucleic acids are used for information storage, transmission, and expression. There's a lot there. Let me break it down. So, every cell on the entire planet, Earth, contains DNA. And the job of DNA is to store the information required to build and maintain cells, or build and maintain an entire organism like you. But that information is stored in nucleic acids, specifically DNA. Now, when cells divide and make new cells, or when organisms reproduce and make offspring, that information needs to be passed on to the new cells. So the new cells know what to do. It needs to be passed on to organisms or offspring to be able to build their bodies. And that's what transmission refers to. So nucleic acids are used for information transmission. The information is transmitted or passed on from cell to cell to cell to cell. And that's been going on for as long as life has been around on Earth, which is at least 3.5 billion years. Finally, what is this information used for? The information is used to make proteins, and that's what expression, information expression refers to. The information is expressed in the form of proteins, because proteins are the worker molecules of cells. Proteins are the molecules that build and maintain cells. Now let me mention to you like an example or analogy of information expression. What I think of is a cookbook that's full of a bunch of different recipes for maybe making casseroles and cakes and salads and appetizers and all kinds of things. And that's what your DNA is like. It's full of a whole bunch of recipes for making structural proteins and enzyme proteins and defensive proteins and all kinds of different proteins. And just like in a recipe book, when we open it up and read the recipe and follow the directions, we put together the ingredients, and whatever food we make is our expression of the recipe. That's how the recipe is expressed. A pie, a cake, a casserole. So in this case, the information is expressed in the form of making proteins, the worker molecules of cells. So that title right there is carrying a lot of information. Now, DNA and RNA are the two kinds of nucleic acids that are found in cells. DNA stands for deoxyribonucleic acid. Quite a tongue twister. And DNA is what's used for information storage. So basically that's where the recipes are. And for information transmission, passing on from cell to cell. DNA consists of separate genes, which are little stretches of DNA that contain the recipes. So each gene is basically a different recipe for making a different protein. The process of copying that genetic information to make a protein is called transcription. And we will get into the nitty-gritty of that in a later chapter. Now, when a gene in DNA is transcribed or copied, the product of that is another nucleic acid called RNA, which stands for ribonucleic acid. And ribonucleic acid basically takes that recipe information in a cell to a place where the recipe information can be expressed and used to make a protein. And that process is called translation, when the information in RNA is decoded and used to make a protein, which is an entirely different kind of molecule. We call that, going from one kind of information to another kind of information, we call that translation. And of course, proteins, like we talked about in part 1, are the worker molecules of cells. They are the molecules that are basically doing everything to keep cells alive. So the figure on the slide is showing you, illustrating some of this, and it shows the flow of information in cells. It begins with DNA, it gets passed on to RNA, and then RNA is used to make proteins. Okay, so in the nucleic acid group of organic molecules, the monomers are called nucleotides. And of course, the polymers are called polynucleotides, and we'll get to that on the next slide. This slide is intended to review what nucleotides are made up of, how can you recognize them, etc. So look at the figure in the top left there of your slide, and it's showing you two nucleotides, a DNA nucleotide and an RNA nucleotide. And you can see some common features to each one. Each one is made of 3 parts: a phosphate functional group, a sugar, and what's called a nitrogenous base. Those are the 3 parts. In DNA and in RNA, all the phosphates are identical. In DNA and RNA, the sugars are slightly different. DNA has a sugar called deoxyribose, and that's because it is a ribose sugar. That's the name of the sugar, ribose, and it is lacking or missing an oxygen atom. So that's where the deoxy comes in, which means without oxygen. Whereas RNA has a sugar that is just a regular ribose sugar, and it has that oxygen in it. Finally, for the nitrogenous bases, DNA nucleotides can have one of 4 different nitrogenous bases, either adenine, guanine, cytosine, or thymine, A, G, C, or T. And if you look in the bottom right of the slide, it shows you some pictures or diagrams of each of these nitrogenous bases. You can see they have slightly different structures. Now, you don't need to memorize those nitrogenous bases, their structures, but you do need to know the different names of them: adenine, guanine, cytosine, thymine. RNA nucleotides also have adenine, guanine, and cytosine, but instead of a thymine attached to some of them, there's a slightly different nitrogenous base called uracil, and that also is shown in that figure in the bottom right. So that overall represents the structure of nucleotides. 3 parts: phosphate group, a sugar, and a nitrogenous base. And it's the nitrogenous bases that are really making the different kinds of nucleotides. And that is really important, as you will see coming up. Okay, the next step is to link together the nucleotides, the monomers, the building blocks, into polymers. And the polymers of nucleotides are called polynucleotides. So all the nucleotides begin with 3 phosphate groups attached to them in cells. Cells build these nucleotides with 3 phosphate groups. And the nucleotides bond together through a reaction called, drumroll, dehydration synthesis. Sounds familiar? I hope so. So when 2 nucleotides bond together, the sugar in one nucleotide bonds to a phosphate group in the next nucleotide. And if you look at the figure in the bottom left, it shows that happening. The figure shows a string of 3 nucleotides already bonded together. There's a phosphate sugar backbone that is forming. That's kind of similar in polypeptides to having an amino carboxyl backbone. You might remember that. You should. Remind yourself. In polynucleotides, it's a phosphate sugar backbone. And you can see on that part of the figure in the very bottom left, 3 nitrogenous bases sticking down, A, C, and T, just for example. And there's a new nucleotide coming on the scene there, and it has a G on it, a guanine on it. And look very carefully and think about what's happening. That phosphate group in the new nucleotide that has the guanine, that phosphate group is bonding to the sugar in the existing chain. And ka-chink, when those link together by dehydration synthesis, you now have a slightly longer polynucleotide, whether it be DNA or RNA. Either way, they both are made up of long chains of nucleotides that bond together, sugar to phosphate, sugar to phosphate, sugar to phosphate, forming a polynucleotide. So you might be able to tell we're kind of building up in complexity here. I'm building up this story. We started off by introducing what nucleic acids are for. Then I started with nucleotides, told you what the 3 parts are, different variations. Then we just left the slide talking about how nucleotides bond together to make polynucleotides, big long chains. Well, here we are. DNA is actually made of 2 polynucleotides, 2 long strings of nucleotides. Look really carefully at the figure that's being shown on this slide, because it shows 2 polynucleotides lined up side by side. Each one of them has a phosphate sugar backbone. And sticking out from each of them are the nitrogenous bases, the A's and the C's and the G's and the T's. And you can see in the figure that the nitrogenous bases are what are matching up. They are sticking together with hydrogen bonds. This makes DNA double-stranded. It's made of 2 strands, 2 polynucleotides, that are matched together with the nitrogenous bases. The nitrogenous bases are always matched up such that an adenine, or an A, in one strand is always matched up to a thymine or a T in the other strand. A guanine, it always matches up to a cytosine. These are called the base pairing rules of DNA structure. A's match T's, C's match G's. This happens for 2 reasons, by the way. One is the size of the nitrogenous bases. There always has to be a big one with 2 rings and a small one with 1 ring. If you look at the previous 2 slides ago on nucleotides, you'll see the 2-ring, 1-ring structure I'm talking about. The second reason for these base pairing rules is the hydrogen bonds that form. A's and T's both need to make 2 hydrogen bonds so they match up to each other, whereas G's and C's need to make 3 hydrogen bonds so they match up to each other. This is what makes the structure of DNA. The final point is that these 2 strands of polynucleotides are upside down compared to each other. And that upside down relationship is called anti-parallel. They are anti-parallel. One is upside down compared to the other. If you look at that figure, you can see that because the phosphates and sugars are flipped upside down in one of the strands compared to the other strand. That is anti-parallel. So to finish this up on this slide, you can really see that DNA has a pretty complex structure. It's a pretty complex molecule. It's made of nucleotides, and the nucleotides are strung together into polynucleotides. And there are 2 strands of polynucleotides that match up to each other, and they follow the base pairing rules. A's match T's and C's match G's. All right, here's the culmination of going through DNA structure. And what we get to is that DNA is made up of two strands, as I've already mentioned, and they twist around each other like a helix, like a staircase, a spiral staircase. And because there's two coils, two helices that are stuck together, that's called a double helix. DNA has a double helix structure. And again, you can see on the figure on the left that in that double helix structure, the guanines in one strand are matching cytosines in the other. G's match C's, and the A's match the T's. So spiraling down through that DNA are these nitrogenous bases that are sticking out like rungs in a ladder, and they're matching up and sticking together, excuse me, not ready to cough, with hydrogen bonds. And that's what's holding this double helix together, are those nitrogenous bases matching up with hydrogen bonds. Now, the final point here is that the structure of DNA lends itself to copying, to making more DNA. And this is what needs to be done when cells get ready to divide and make more cells, is that information, that library of information that has all the genes, all the recipes for making the proteins of your cells, that information has to get copied so that each new cell that comes from an original cell will get a complete set of information, will get a complete copy of DNA. So what happens is the two strands that make up DNA pull apart. They literally separate. And each strand is used as a template to make a new strand. We're going to probably cover this later in this class. So if one strand pulls apart and it has an A on it, then a new nucleotide with a T will come in and match up to it, and ka-chink, it'll start making a new strand that matches up to the original one. Now the other strand that pulled apart has a T in it, so an A would come down and match up to it, and ka-chink, you'd have an A and a T. And if that keeps happening with each of the original template strands, new nucleotides coming down and matching up to them, we will build two new identical DNA molecules. Again, we may get to this later in the class, but the overall point here is that the structure of DNA lends itself or allows it to be copied to make more DNA. And this is important for passing on information to new cells. So we're wrapping up this nucleic acid category by briefly giving an overview of what's called gene expression. Remember, the first slide in this PowerPoint or this video talked about how nucleic acids are used for information storage, transmission, and expression. So this is talking about the expression part. This is like taking a recipe in a cookbook and expressing it by making a cake or a casserole. So it's pretty complex. We will definitely go over the nitty-gritty of this in a later chapter, but this is just giving a brief overview. So try to wrap your brain around this introduction and remember it. It'll come back. So the DNA has these different stretches of recipes in them, and those different recipes are called genes. In order to make a protein, a gene in the DNA has to be copied. And the copy of that gene is the other kind of nucleic acid called RNA. Basically, the DNA unzips and the recipe is exposed. And RNA nucleotides come down and match up to the DNA recipe, and the RNA that forms by matching up to the DNA, that RNA becomes a copy of the DNA. And then that copy, that RNA messenger copy, leaves our nucleus of our cells and goes into the cytoplasm, and that's where that genetic information is used to make a protein or polypeptide. Now, because RNA has information in it, the information is in the sequence of the A's and the C's and the G's and the U's. That's how the information is stored. That's basically a language. That's a molecular language. And that language in that RNA is used to make a sequence of amino acids in a polypeptide, which is like a whole different language. So here's the crux of it. Because we're beginning with RNA language and using that to make a polypeptide, a different language, the process of doing that is called translation. The message in the RNA is being translated into a polypeptide. So that's what you basically need to know for now. We will come back to nucleic acids in a later chapter to learn very specifically how they are used to make the proteins of your cells. But this gives you a little package to understand the structure of nucleic acids, how they're put together, and what they're used for. Information storage, information expression. All right, moving on to the carbohydrate category. When you hear, think, read, dream carbohydrates, think sugars. Carbohydrates are the sugars. Now, just like in the protein category where there's monomers and polymers, just like in the nucleic acid category where there's monomers and polymers, the same thing applies to the carbohydrates. The monomers, or the building block molecules, are called monosaccharides. And those are called simple sugars. Mono means one, saccharide means sweet, so it's referring to sugar. So those are the building blocks. And if a whole bunch of monosaccharides are linked together, bonded together, then we have polysaccharides, many sugars. So there's an intermediate category too of sugars called disaccharides, and that's when two monosaccharides link together, ka-chink, and you have a slightly bigger molecule that's called a disaccharide, which means two sugars. We'll take a look at those coming up. But these are both called sugars, mono and disaccharides, because they taste sweet. Polysaccharides don't taste sweet to our tongues, so we don't call them sugars. There's two main functions of carbohydrates in cells, living things. One of them, the primary one, is energy storage. This is the main way that cells are storing energy to use it later on, to build things or move things or maintain things. The other main function or job of carbohydrates is for cell structure, for holding things together or building things. So you'll see a couple examples of this. Before we go, I put a photo on the slide here to give you some sense of what kinds of foods are rich in carbohydrates, rich in sugars. So you can see potatoes, they're full of starch, which is a polysaccharide. You can see pasta, which is also full of starch, a polysaccharide. Breads, full of starch. But you can also see like honey, which is made up of mono and disaccharides, and it's more sweet. Examples of fruits, bananas, dried apricots, etc., which have sugars in them. So soak up that image so you can think to yourself, these are some of the foods that have lots of carbohydrates in them. Just like we did in the protein group, where we first looked at amino acids, how they're put together, and we did for the nucleic acid group, where we first looked at nucleotides and how they're put together. We're starting off here by looking at monosaccharides and how they're put together. So here's a figure of several monosaccharides that are very common in your and my bodies and cells of other creatures. And you don't need to memorize the structures of these, but the notes I have here and that I'm talking to you about, you definitely need to be familiar with this information. So, number one, monosaccharides have a molecular formula that is some multiple of CH2O. So, they have three different elements in them: carbon, hydrogen, and oxygen. And for every carbon atom, there are two hydrogens and one oxygen. 1 to 2 to 1. 1 to 2 to 1. That's the ratio. Secondly, monosaccharides have lots of hydroxyl groups, which, by the way, makes them polar and easily dissolve in water. And they have either a ketone or an aldehyde carbonyl group. Remember those from earlier in the chapter? Carbonyls are double bonded oxygens to carbons, and if you look at the figure I'm giving you here, the carbonyls are color-coded with kind of a salmon orange color. The aldehydes are at the ends of the molecule, and the ketones are somewhere in the middle of the molecule. This is very consistent. It coincides with what I told you about aldehydes and ketones in part 1. Thirdly, monosaccharides have carbon chains typically with 3, 5, or 6 carbons. Those are called triose, pentose, or hexose sugars. Ose means like sugar, tri, pent, and hex means 3, 5, and 6. With all this information in mind, now take a closer look at the monosaccharide figure that I put on your slide. If you look in the left-hand column, it says aldehyde sugars. Those are all sugars that have aldehyde groups on the end. And glyceraldehyde has 3 carbons, and look at it. For those 3 carbons, there are how many hydrogens? Count them up. There are 6, twice as many. And how many oxygens? Count them up. There's 3. There's your ratio, 1 to 2 to 1. If you look down below at ribose sugar, that's a pentose. It has 5 carbons. And once again, you can see the 1 to 2 to 1 ratio. 5 carbons, 10 hydrogens, and 5 oxygens. You can look at all the rest of the sugars that are shown here, and you'll see the same pattern. You can also see lots of hydroxyl groups on these things. So let's move on. We're going to see how monosaccharides are linked together to make bigger molecules. All right, there's 2 main points on this slide. One main point is that if you have a little teaspoon of sugar, monosaccharides, those sugars are going to be linear. They're going to be straight, kind of like the figure on the previous slide. Those molecules are straight. And that's shown in the top left of this slide. But when those sugars are placed in water, when they're surrounded by polar water molecules, those linear molecules wrap around and form a ring. And you can see that in the top left corner of the slide. It shows the linear molecule folding around and turning into a ring. So all the sugars that exist in cells, which are watery environments, exist as rings. And when you see pictures of sugars in a biology textbook, they're almost always drawn as rings because that's how they exist in the watery environments, the aqueous environments of cells. So the other major component of this slide is to show you how monosaccharides bond together to make disaccharides. And if they kept bonding together more and more, you'd make a polysaccharide. So look carefully at the figure on the right-hand side of the slide. It shows 2 glucose molecules in figure A side by side. And just like you learned earlier in the chapter, a hydroxyl, an OH, is taken from one, a hydrogen is taken from the other. They bond together, ka-chink, and make water. That's dehydration. And then ka-chink, the 2 monomers bond together and you have a bigger molecule. In this case, the 2 monosaccharides bond together and you have a disaccharide. If 2 glucoses link together, that makes something called maltose. In part B, or figure B, if a glucose and a fructose bond together, those make sucrose. And sucrose is a very abundant sugar in plants. And that's what mostly tastes sweet to your tongue when you eat a fruit, it's the sucrose. So dehydration synthesis is linking those monomers, those monosaccharides together to make bigger molecules. Yes, now plants transport sucrose from the roots to the fruits. No, that doesn't make sense. The sucrose is made in leaves in photosynthesis. That's where it's made. And then it's stored in fruits. So mix that up. In mammals like us, lactose is a disaccharide that's found in milk. And that's what female mammals, mothers, in part feed their babies when they feed them milk, is lactose. And lactose is a very high energy molecule. It's providing energy to a newborn or baby mammal. Okay, well, I didn't do this for nucleic acids, but I threw in a slide here, called it Concept Check, for you to answer some questions and kind of review what I've talked about so far for carbohydrates. This is probably a good thing to do. So anyway, I've got these questions on your worksheet. So look these questions over. And again, these are review questions. I've talked about this. So see if you can answer them from your memory. And if you're having trouble, go back and look at your notes and you'll be able to find the answers. But I'll leave it at that. Just kind of a little review for you. Test questions. Okay, well, I pulled this in from outside of your textbook. I don't think your textbook really touches on this subject so much per se, but I figure it's worth sharing with you because so many Americans ingest or eat too much sugar, and it's very bad for us in many ways. So everyone should be moderating or decreasing their sugar consumption. Many of our foods that we eat, that we purchase in supermarkets, are loaded with sugar to make them taste sweet, to encourage us to buy them and eat them. But it ends up killing us, or could anyway. So let's take a look at how Americans' average sugar intake compares to what is recommended by the World Health Organization, WHO, and the Federal Drug Administration, Food and Drug Administration, FDA, which is a United States government administration. So Americans consume over 6 times the amount of sugar that the World Health Organization recommends, and over 3 times what the Food and Drug Administration recommends. So right there, I'm letting you know, we're eating overall too much sugar. And the other overall point I want you to learn here is what are some of the health consequences of eating too much sugar? Well, we've got a list there. Dental cavities, your teeth can rot. Obesity, having too much fat on your body. 37% of adults in the United States are obese. That leads to many different health complications as one gets older. Type 2 diabetes, which is a horrible disease. And high blood pressure, which can lead to heart attacks or strokes and kill you. So, sugar, while it's important, we all need it. All living things use it and need it. Americans tend to consume too much. Be careful with your sugar intake. All right, well, let's now get to the level of polysaccharides. We've talked about monosaccharides, disaccharides, and now we're to the polymer group called polysaccharides, which means many sugars. So let me first talk about the information on the bottom of the slide, and there's information up on the top right too that goes along with the figure, top, middle, bottom. So there's a lot of information on this slide about storage polysaccharides. Now, what's meant by storage is these are polysaccharides that living things use to store energy, to pack it away for later, to use it later. Now, first, polysaccharides are long chains, polymers, of the building blocks called monosaccharides. So they're bonded together one after another, ka-chink, 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 with dehydration synthesis reactions, and that makes polysaccharides. These are used, the examples on this slide, for energy storage, or the next slide, I'll show you examples, they're used for structure, holding things together. Those are the 2 main functions of polysaccharides: energy storage and structure. In the plant world, starch is the polysaccharide that's used to store energy. And in the animal world, glycogen is used to store energy in the form of sugar. And both of these polymers are hundreds to thousands of glucose molecules long. Look at the figure that I've got here. The little diamond-shaped emblems, if you will, are the sugars, the glucoses, and you can see lots of them linked together into long chains. So those glucoses are being linked together to make either starch or glycogen. Cellulose is also shown on this figure, but cellulose really belongs on the next slide because it's a structural polysaccharide that we find in plant cells to make the plant cells strong. So let's stick to starch and glycogen on this slide. Both of them are helical, which means that they're coiled, okay? And you can see in the very upper picture, the guy's holding, or somebody's holding some potatoes, and those potatoes are full of starch. They're full of lots of sugar energy, coiled starch molecules. In the meantime, this person's muscle cells are storing glycogen, and those are also coiled, and they have little branches sticking off of them. Both starch and glycogen are full of energy that organisms are storing for later use. Plants are storing their starch in their roots, a little bit in their stems. Seeds get starch to feed the embryos, and whenever the plants need a sugar source, they hydrolyze or break down those starch molecules into glucose and then use the glucose as fuel. We do the same thing. If you eat a meal, you're going to take all the extra sugar that you don't need at the moment. It goes from your digestive system into your blood, and then your blood brings it to your muscles and your liver. And in your muscles and in your liver, the extra sugars that you don't need right away, the extra glucoses, are linked together to make glycogen. And the glycogen is stored in your muscle and in your liver tissues, and it's used within about 24 hours. So it's slowly broken down over 24 hours to put into your bloodstream to basically feed the cells of your body. So plants are storing starch kind of long term. We as animals are storing glycogen in the short term. And if you don't have another meal within a day, then your glycogen pretty much gets depleted. You use it up. All right, the last slide talked about polysaccharides, what they are, being made of glucose mostly, and particularly it focused on storage polysaccharides, starch and glycogen for storing energy. Well, other polysaccharides are used for structure, for holding things together. And probably the most important one for you to know is cellulose. Cellulose. Cellulose is found in the cell walls of plant cells. The outer boundary, the outer framework of plant cells is made up of cellulose. And the cellulose forms rope-like structures. They're really strong. Each cellulose molecule sticks to other cellulose molecules nearby with hydrogen bonds, and by all these long cellulose molecules sticking together with hydrogen bonds, it makes strong rope-like fibers. They're called microfibrils. If you look at that figure I put in the upper left, you can really see all these things. There's a plant leaf. That plant leaf, that maple leaf, is made of thousands and thousands of microscopic cells. Each one of the cells has a cell wall. Check that out in the figure. If you blew up and magnified what's in the cell wall, you would see that it's packed with these microfibrils, these little rope-like structures. And if you then magnified even more, you'd see that these microfibrils are made up of individual long cellulose molecules that are sticking together with hydrogen bonds. So that's what makes plants strong and tough and helps them to stand upright on land, is the cellulose in their cell walls. Cellulose is the most common, abundant organic compound on Earth. Plants are constantly making sugars through photosynthesis, and they're linking those sugars together to make cellulose to hold up their bodies. 10 to the 14th kilograms of cellulose is made every year by plants on Earth. Pretty amazing. Now, notes on the bottom here, most animals cannot hydrolyze or digest cellulose. We do not have the enzyme that'll break apart the glucose molecules and digest the cellulose. Bacteria and funguses do make some bacteria make an enzyme called cellulase. And that cellulase is able to break down the cellulose into individual sugars, and then the organisms can use those sugars for energy. In us, we can't digest that cellulose, and so it passes right through your digestive system and comes out in your poopy. And that is what we refer to as insoluble fiber. And that fiber is really important to your health. It scrapes or abrades against the sides of your intestines, stimulating the release of mucus, and that helps your food pass through your intestines. So the message to you is not only have a diet that lowers the amount of sugar you're eating, but also have a diet that has lots of plant material in it, lots of fiber in it from especially vegetables, because that helps keep your intestines healthy. You won't get constipated as much, and it could even help prevent cancer, like colon cancer. So how do plant-eating animals survive if they can't hydrolyze or digest cellulose? Like cows and goats and camels and deer, they're all eating plants. That's all they eat. And I just told you that animals can't digest cellulose in plants. Well, they have bacteria that live in their guts, in their stomachs. And the bacteria break down the cellulose that those animals are eating. And then the animals like goats and cows and camels have access to the sugars once the bacteria break them down. Pretty cool. Last point here, there's a polysaccharide found in arthropods like insects and spiders, scorpions, and it's called chitin. And it's in their exoskeletons. It's in their outside skeletons. And that's partly what makes those creatures go crunch when you step on them, is that chitin is a tough, strong polysaccharide. Yeah, it's also found in fungi. We, as humans, have learned to use this chitin technologically to make surgical threads that you could tie up a wound or make a suture in a patient after surgery, and that chitin will break down on its own, and those threads will break down on their own, and you don't need to go back into a doctor's office and have them physically move the threads like surgeons or doctors used to have to do. So that's kind of neat. So let's move on to the last category out of 4 of organic molecules. We've covered proteins, nucleic acids, and carbohydrates, and now we have made it to the lipids. And as the title says here, lipids are a diverse group of hydrophobic molecules. There are many different kinds of molecules structurally that are put into the lipid category. And what they all have in common, the reason why they're all put into this category, is because they are nonpolar, therefore they are hydrophobic. Lipids include some familiar kinds of molecules like fats, steroids, waxes, pigments, and less familiar to most people, an extremely important type of lipid called phospholipids. Now let me touch on the nonpolar part. Remember from chapter 2 that covalent bonds are nonpolar if the atoms that are making the bond share electrons equally. And the two biggest candidates that do this are carbon and hydrogen. Carbon's electronegativity is 2.5, hydrogen's is 2.1, the difference is 0.4. And because the difference is so small, their covalent bond is nonpolar. And when you have molecules that are made up almost exclusively of carbons and hydrogens, then all those covalent bonds are going to be nonpolar, and that's going to make the overall molecule nonpolar, and it won't mix with water, and that's what makes it hydrophobic. Now, the lipid category does not include true polymers. You might remember from the early part of the chapter when I talked about the different kinds of organic molecules and their monomers and polymers, I specifically said lipids don't have a specific kind of monomer. So there's no replicating monomers. And they're not really big enough to be considered macromolecules like proteins or nucleic acids or polysaccharides. I mean, they are fairly big molecules, but they don't even come close to some of those others. So just for some examples, I put some pictures on the slide and identified them. So vitamin D, for example, is a lipid. You can see it looks kind of like a steroid. In fact, it is a steroid-based molecule based on the rings. We'll get to that. Xanthophyll, that is a pigment that makes plants look yellow. Like when you see leaves that are yellow, that is a pigment for capturing sunlight, and that's called xanthophyll. Look at all those carbons and hydrogens. And then finally on the bottom, there is a wax ester. That's what it's called. And that's what covers the leaves of plants, helps make them waterproof, covers insects, makes them waterproof, mammals. And that kind of zigzagging structure you see is the chains of carbons and hydrogens. So at every one of those zigs and zags is a carbon, and then there's two hydrogens sticking out. So rich in carbon and hydrogen. You can see right in the middle of that wax ester, there's a ketone. There's a double bonded oxygen, and then an oxygen in the middle there. So anyway, those are some different kinds of lipids. Let's take a closer look at some of these different kinds. So we're going to begin with fats, and fats are called triglycerides. Tri because they have 3 fatty acids, and those 3 fatty acids are bonded to a little molecule called a glycerol. It's a type of alcohol. So that is the definition of a fat. A glycerol bonded to 3 fatty acids. Now let's quickly review what a fatty acid is. Look at the figures and listen to and read my descriptions. Fatty acids are hydrocarbon chains with a carboxyl group on one end. And usually those chains are 16 to 18 carbon atoms in length, but there are many different sizes of fatty acids, different types. So hydrocarbon chain, that is simply what it sounds like. A chain of carbons with hydrogens attached to them, hydrocarbon. And if you look at that figure on the left, it shows you the carboxyl group on one end. It's kind of highlighted in blue. And that carboxyl group is going through a dehydration synthesis reaction, should not be a surprise, to connect to that glycerol. And ka-chink, you get a connection. And with 3 of those fatty acids making what are called ester linkages, the kind of bond between a glycerol and a fatty acid, you get a fat. Now, the major function of fat is energy storage. It's a very efficient way of storing energy because all those carbons and hydrogens, the bonds that form between the carbons and hydrogens are rich in electron energy. And that's where the energy is. And fats have a lot of them. In fact, if you compare a gram of carbohydrate, sugar, to a gram of fat, the gram of fat will have twice as much energy in it. That's why animals store their energy as fat. That's why we mostly have fat on our body and we're not covered in starch like plants, because fats are a more efficient way to store energy, so we don't have to walk around or fly around or swim around with as much energy storage molecules being part of us. But fats do more than just store energy. They also help to cushion vital organs. So organs are surrounded by fat, such as around your kidneys. And of course, right beneath your skin, there are layers of fat, and that fat helps to insulate your body and help keep warmth inside. And I tried to write on the bottom and it didn't come out, used to make hormones. So fat molecules are used to make basically steroids, hormones. I have an image, you can see there, a photo on the bottom corner of the slide showing you some different kinds of foods that are rich in fat. So dairy, butter, milk is naturally rich in fat. Meats, like red meats, like beef. Oils from plants, oils are fats. And fish, some fish are particularly fatty, like salmon. And so what we're going to cover next is what's the difference between a saturated fat and an unsaturated fat. So let's take a look at that. Well, you may have heard before that saturated fats are bad for you. Unsaturated fats are not as bad for you. So first of all, you're going to learn here what makes a fat saturated or unsaturated. And then you're going to learn in a little bit as to why one is healthier than the other. So here's the thing. Saturated fats do not have any double bonds between the carbon atoms in their chains. So all the carbons, because they all have single covalent bonds between them, they are all able to bond to two hydrogens. So every carbon in that chain is saturated, is filled with as many hydrogens as they can possibly bond to. So you can see figure A, it's showing you a saturated fat molecule in butter, and that zigzaggy line is the carbons, bonding together in a chain, and there's two hydrogens coming off of every carbon. And those saturated fatty acids that are straight, they're straight. And the fat molecules basically are all flat because all the fatty acids are straight. And so saturated fat molecules stack together like pieces of paper. And because they stack together so evenly and compactly, saturated fats are solid, not liquid, solid at room temperature. So that's why you can put a stick of butter out on a countertop and basically at room temperature, like 72 degrees Fahrenheit, your butter is going to remain solid. All those fat molecules are packed close together. Unsaturated fats, let's cover that. Unsaturated fats have at least one fatty acid out of the three that has a double bond, one or more double bonds in the carbon chain. And any place that there's a double bond in the carbon chains, that makes the fatty acid become bent or kinked. So unsaturated fats, the reason why they're called unsaturated is because if there's two covalent bonds between two carbon atoms, each of those carbon atoms can only bond to one hydrogen, not two. So those carbons are not saturated with hydrogen, they are unsaturated. That's why they're called unsaturated fats, because the carbons are not saturated with hydrogens. Now, unsaturated fats don't stack together neatly like pieces of paper, but because the fatty acids are bent or kinked, they're kind of in a jumbled mess, kind of like crumpling up pieces of paper into little balls, and you try to stack them, they're not going to stack nicely, you know. And so unsaturated fats, because they don't stack so neatly at room temperature, meaning like 72 degrees Fahrenheit, they're liquid. They're a liquid state. And they have to be much colder than that to become a solid. So unsaturated fats are liquid at room temperature, and they are what we call oils. Oils are unsaturated fats that are liquid at room temperature. So think of canola oil, or corn oil, or olive oil, peanut oil, safflower oil. These are all unsaturated fats that are liquid at room temperature, and they come from different plants. They are oils. Now I'm going to talk about cis fatty acids and trans fatty acids. And I have another slide that talks about this, because cis fatty acids are bent. That's the natural kind of fatty acid, and they're healthy for us. Trans fatty acids are straight, and they're very unhealthy for us. In fact, your nutrition labels are required by law to tell you how much trans fat is in your food because it's so bad for you. So hopefully you're understanding the difference between saturated and unsaturated fats. And let's do a little bit more. Okay, I'm telling you a few things about essential fatty acids here. Remember, fatty acids are some of the components that make up fats. Essential fatty acids are ones that you have to eat in your diet, in your food, because your body is unable to synthesize or make them. And you need them. You need them as part of your healthy biology, but you have to eat them. And so anything that you have to eat, get from a food source because your body can't make it in the nutrition world is called essential. So omega-6 and omega-3 fatty acids are both essential fatty acids, meaning you need them and you have to eat them. Now omega-6, that just simply means that the double bond, the kink, is at the number 6 carbon in the chain. So if you look in the figure I've got on the left, it shows linoleic acid. It's omega-6. And if you start on the far left part of that fatty acid and count over 6 carbons, you'll get to a double bond. So that's an omega-6. In fact, you can see 2 double bonds in that fatty acid, so there would be 2 kinks in there. It would be pretty kinky. Alpha-linolenic acid is an omega-3. And so if you count from the left, 1, 2, 3 carbons, you get to a double bond. That's omega-3. And there's a couple other double bonds in there, so that one's really kinky. Foods that are rich in these fatty acids, essential fatty acids, these omega fatty acids are pictured here. So eat some fish like salmon, olive oil, legumes like beans and peas, kale. So there's several foods that you can get your essential fatty acids and be healthy. All right, here I'm going to tell you about hydrogenated oils. Hydrogenated oils. If oils are hydrogenated, let's see, the process is called hydrogenation. And you can look at ingredients of many foods, or if you buy margarine in a supermarket, you will see listed in many foods hydrogenated fat. That means that they took an oil from a plant, a vegetable oil, and they added hydrogens to it. Now what that does is the double bond between 2 carbons goes away, and the 2 carbons are then only linked by a single bond. And when they get linked by just a single bond, then the carbons can hold 2 hydrogens, not just 1 hydrogen. So that's hydrogenated. Hydrogens have been added. The reason why food Manufacturers make hydrogenated oils. The reason why they take normal oils that are unsaturated and hydrogenate them is because they become more stable. They don't break down and get rancid or what's the word I want? Spoil very quickly. So that's a big reason. And also because they're more solid, they're less messy. You know, so you pick up an Oreo cookie that's got hydrogenated oil in it. It's more stable and it's less messy. You don't have oil like dripping out of the cookie. It's more like butter. So that's why oils are hydrogenated. But problem is, hydrogenated oils are not healthy for you. They're not very good for you, kind of like saturated fats. So you should try to minimize the amount of food that you eat with hydrogenated oils. It's not good for you. It might lead to heart disease. It might lead to your arteries clogging up and getting blocked and giving you a heart attack, which I don't think any of us want that. So take a look at the figure I've got here on the page. So the top part is showing you an unsaturated fatty acid. It says unsaturated fat, but it's just showing you a fatty acid, and it's got that double bond in there. So if that double bond is taken out, and now there's just a single bond between the carbons, they can now get two more hydrogens on them, and the molecule straightens out, and okay, we like that in our foods a little better, a little cleaner and a little more stable. So that's hydrogenation of oils. Now, I told you a couple slides back I was going to talk about or mention trans fats because they're so bad for us. I almost certainly grew up eating foods with lots of trans fats, not good for me. The good thing is that food manufacturers are now required by law to tell you if their foods have trans fats and how much they have. Not many food manufacturers are putting trans fats into their foods. They want people to buy their foods. So you don't have a lot to worry about. So what happens is that you start off with a regular fatty acid, and look at that picture on the bottom left corner of your slide. See that there's a double bond between two carbons and they're blue. Well, typically what happens in natural oils is the hydrogens are coming off on the same side of the fatty acid. And that arrangement of hydrogens coming off on the same side is called a cis, C-I-S, a cis fatty acid. And it's bent or kinked. In the process of hydrogenating oils, some of the fatty acids keep their double bond, but they become trans fatty acids. And what happens is one of the hydrogens moves to the other side of the fatty acid. And now one's pointing up and one's pointing down. Look at that on the bottom of the figure. And when that happens, the fatty acid becomes straight instead of kinked or bent. And then that makes it less healthy. That makes it act more like a saturated fat, which is bad for you. And that's part of the reason why trans fats are so bad for you, because they get straightened out and they can clog your arteries, give you heart attacks. In fact, trans fats are worse for you than eating saturated fats like you get from eating meats high in fat or dairy with a lot of fat. So as I said, we don't find many trans fats in our foods because food manufacturers make sure they leave them out. But what I want you to know here is what is a cis fat versus a trans fat. Okay, so look at the figure and know the difference in health consequences. Okay, we're finished with the fat category. Now we're going to touch on phospholipids. And in another chapter or so, we're going to start talking about cell structure. And a majorly important part of cell structure are the membranes of cells, the parts of cells that form the outer layers and boundaries. Cell membranes are really important. And we're going to be covering more about how they work and what they do and why they're important in an upcoming chapter. But for now, you need to know what phospholipids are, because phospholipids make up our cell membranes. So here you're just learning what phospholipids are, what they look like, and what they're used for. So a phospholipid is fairly similar to a fat because it's made of one glycerol, but instead of 3 fatty acids, it's made of 2 fatty acids. And instead of the 3rd fatty acid, in its place, a phosphate group attaches to the glycerol with a nitrogen group called a choline group. Look at the figure in the upper left. It's showing you a couple different ways of picturing a phospholipid. Okay, if you look on the far left, see where it says hydrophobic tails? Those are the 2 fatty acids. And if you look at that picture, you can tell in the picture one of them is saturated, it's straight, and one of them is unsaturated, it's bent or kinked. That's how most phospholipids are. Not all, but most. Those are called hydrophobic tails because they look kind of like tails, and because they're non-polar, being made of carbons and hydrogens, they're hydrophobic. Now the other part of the phospholipid is called a hydrophilic head, and it's made of the glycerol with the phosphate and the nitrogen group. And that whole part of the phospholipid is charged, has an electrical charge, like that phosphate group is negative and the nitrogen group is positive. So because it's charged, it's attracted to water, and that's what makes it hydrophilic. Now look in figure C, look at part C. That is the symbol that we use in biology for a phospholipid. And you should be familiar with that symbol. A little oval is the hydrophilic head, and the 2 little yellow lines sticking down are the hydrophobic tails. So you should be able to draw that symbol and label hydrophilic head, hydrophobic tail. If you look in the very bottom of the slide, it's showing you how phospholipids arrange themselves to form our membranes. Look how there are 2 layers of phospholipids that are all lined up. Membranes, the structure of the arrangement of phospholipids in a membrane is called a bilayer. Bi means 2, so 2 layers. You have 2 layers of phospholipids. And the reason why membranes have 2 layers of phospholipids, it has to do with the hydrophilic heads and tails. In one layer, the hydrophilic heads are pointing towards water, like on the outside of a cell, say, a living cell. And on the other layer of the membrane, those hydrophilic heads are pointing towards water, like on the inside of the cell. And so with water on both the outside of a cell and on the inside of a cell, that just naturally makes 2 layers of phospholipids line up with their hydrophilic heads pointing out and the hydrophobic tails kind of huddling inside, hiding from the water. So we're going to get into a little bit later just how the structure of membranes like this is really important for controlling or regulating what goes in and what goes out of cells. The phospholipid membranes of cells are kind of like the gatekeepers that control what kinds of molecules and ions can go in and out of cells. It's a really important function of membranes, is controlling what goes in and what goes out. But from the slide, you should recognize the structure of a phospholipid: glycerol with 2 fatty acids and a phosphate group and a choline group. You should know that those fatty acids are called hydrophobic tails, and the glycerol and phosphate group end is called a hydrophilic head. And you should know that phospholipids line up in 2 layers called a phospholipid bilayer when they form membranes of cells, because of water being on the outside and water being on the inside, that forces them to make that bilayer. Okay, we're wrapping up lipids, and in fact, we're wrapping up this whole chapter on organic molecules with steroids. Steroids are also lipids because, I could ask you why, they're full of carbons and hydrogens and they're nonpolar, therefore they are hydrophobic. So steroids are lipids. Now you can recognize in a picture a steroid because they are made up of 4 carbon rings that are all attached together, like the pictures I'm showing you here, with various functional groups sticking out. And that's what makes the different kinds of steroids are the different functional groups, the type, the number of them, and the placement of the functional groups. The steroid that's shown in the upper left, in fact, is cholesterol, a very important molecule for cells and for living things, cholesterol. Not only is cholesterol a component of cell membranes, it's kind of interspersed among the phospholipids and helps the membranes maintain a fluid structure. But cholesterol is also used as sort of a starting point for making hormones in your liver, hormones that are used as messenger molecules in your body to send chemical messages. And that's what hormones are. Hormones are messenger molecules, and they are steroids. Now, high cholesterol contributes to atherosclerosis, which is the hardening of your arteries. If you have too much cholesterol in your blood, it can lead to what are called plaques that form inside your blood vessels, and it's like a bunch of fat or crud sticking together and slowly but surely clogging the inside of your blood vessels so blood can't get through. And if that's going on in your heart or in the arteries coming out of your heart, if they're getting clogged by plaques from cholesterol, your heart's got to pump harder and harder and harder to push the blood through. And eventually, if your heart doesn't get enough oxygen because it's getting clogged up, you could have a heart attack. So high cholesterol, another bad thing. Watch the amount of cholesterol in your diet. On the other hand, cholesterol is really important. Yeah, you've got to have it to be healthy. So like everything else, you need moderation. You need a balance. Now, look in the bottom right of the slide. I'm simply showing you some different hormones. And so they all are, like I mentioned, are different based on their functional groups. But what they have in common is that 4-ringed structure. So whether you're cholesterol, estradiol, testosterone, progesterone, cortisol, aldosterone, those are all different hormones. They all have a very common and similar structure.